Hello everyone, welcome to Get My Class. Let's solve the question. It says PCR uses which of the following? Option A, DNA polymerases, deoxynucleotides, DNA oligos. Option B, DNA polymerases, deoxynucleotide, DNA oligos. Option C, RNA polymerases, deoxynucleotides, RNA oligos. Option D, DNA polymerase, deoxynucleosides. Option D, and the last one is DNA oligos. So let us understand the question. It says PCR uses which of the following? So first we have to understand what do you mean by PCR. What is PCR? PCR is nothing but polymerase chain reaction. Okay, what is polymerase chain reaction? Polymerase chain reaction is a technique where you can say that this is used for amplification of DNA. Okay, DNA, it can be just a small fragment of DNA or a particular gene, you can say. Okay, that is what is utilized for. That is polymerase DNA chain reaction is utilized for amplification of DNA. So when we say amplification of DNA, there will be, you'll be having the sample, that is the template strand you have to provide. Okay, and further the template strands will be used as template and new strands will be synthesized. Okay, so DNA template strand will be provided. DNA template strand will be there. Okay, DNA template strand. Using this as template, new strand will be synthesized. Now, for the synthesis of new strand, what are required? Of course, you need DNA polymerase, right? What is DNA polymerase? Polymerase is nothing but it is an enzyme which has the capacity to extend the nucleotides, to add the nucleotides and to extend the strand. Okay, and one of the important DNA polymerase here which is used is for a stack polymerase. So what is stack polymerase? Stack polymerase is a thermostable polymerase because this reaction there is a high temperature at 70 degree Celsius, you can say, around where stack polymerase can survive, and that means it can be active. So it is an enzyme, so it can be active and perform the extension of the strand, that is addition of the nucleotide. Now you have the enzyme. What else you need? Of course, you need the nucleotides, right? You need the nucleotides, that is in the form of DNTP. Okay, that is deoxyribonucleotides. So here the nitrogen base can be in the form of adenosine, then ATGC you can say. Okay, thymine, guanine, and cytosine. Okay, so these are all what? Nucleotides, that is nitrogen base containing nucleotides. So of course you need DNTPs, okay, deoxynucleotides. Then of course you also need short stretches of DNA, which is called as DNA oligos, okay, which act as the primer, These are the primers. Okay. Now primers are necessary because primers are the one which will be added and they will provide the 3' free end for the addition of next nucleotide. Okay. So that is very important here. And that is how, suppose you have the DNA, this is the template DNA. The primers will first attach. Okay. These are called as oligos, and further extension will be done by the polymerase. Okay, because polymerase can add only to the 3' hydroxy end. Okay, and synthesis of what? In 5' to 3' end, right? That is how the synthesis occurs. And that's why you need DNA oligos. So let us see the option. Option A says DNA polymerases. Yes, it is required. And dideoxynucleotide. No, we don't need dideoxy. Okay, we need only deoxynucleotide. Hence, it is ruled out. And here it says DNA oligos. Yes, we do need DNA oligos, but not dideoxynucleotide. We need deoxynucleotide. So it is ruled out. Option B says DNA polymerase. Yes. Deoxynucleotides. Yes, we do need. And DNA oligos. Yes. DNA oligos, please remember, it acts as a primer. So this should be the correct answer based on our explanation. Option C says RNA polymerase. So we don't need RNA polymerase here because here we are using DNA oligos, so we don't need RNA polymerase. And hence this totally is ruled out. Option D says DNA polymerase. Yes. Deoxynucleosides. So we don't require nucleosides, we need nucleotides. So there is difference between nucleosides and nucleotides. Okay, you have to keep that in mind. And here this says DNA oligos, that is correct, but not nucleoside, it is nucleotide. Hence this is ruled out. So what is the correct option? The correct option is option B. I hope you understood the explanation. Thank you, everyone.